All right, here we are at South Dakota State University in the ICECL lab. That's right, they study ice here. And in this freezer, it's negative 20 degrees Celsius. And we've got samples of ice that are over 500 years old. Here we go. Here we go. Here's the ice cores right here. Here's the ice cores right here is where they work on it. Okay, here's our little chunks of them. And then over here, we've got two different ways to analyze these ice cores. The first way is you actually just cut them up into chunks and then let them melt and then analyze the water that comes back. To cut them up, they've got just a regular bandsaw. How crazy is that? A regular bandsaw cuts them up, cut them up into little chunks while you're freezing out here, negative 20 degrees. And then this guy has a new contraption they've got set up. It is called a melter. Okay, so they put that ice core, once they've cut it up into a strip, they put it in here, and it goes down here and melts, and then it travels through this tube, where it'll put it into different sample vials. We'll look at that. So here we are. Remember that tube that went through the wall from the melter? It comes out right here. That's where that tube comes out, okay? They got a lot of tricky things to worry about. Remember, it was negative 20 degrees. I'm back here in a regular room temperature room, but there's a lot of things to worry about because out in that cooler, it's negative 20 degrees. That water will freeze almost instantly. So they have to use insulation and heaters just on the tube in order to keep it from refreezing after they've melted it. And that's why it's got that black insulation around that tube. And then that water is gonna travel through there into these two tubes. And they have two different tubes that come out. One is the outside of the ice, that sample that they cut, and they keep that separate from the inside of the ice because they're really, really worried about contamination. Remember, that ice has been under the surface for 500 years, so they got to make sure nothing gets in there, absolutely nothing. So they want to make sure that as much as they possibly can, that the ice that they're studying is the ice that's been there for 500 years. So right here, they take the one side, see this one's labeled waste, and it comes out here and they put it over here. Now they're doing a special experiment where they actually want to test it to be able to contain, or to be able to figure out which ones are being, how much contamination is actually happening. And then over here, this tube will run over here and drop it in the test tubes. They don't have any vials set up right now, but these are called auto samplers. And it's gonna just automatically grab whatever comes out. So they can go through, instead of cutting each individual chunk and measuring it, they can actually, they can say, okay, we want to take 5 milliliters at a time and just record 5 milliliters, and this machine will actually capture it and keep track of it as it goes. So they can run meters and meters of ice cores all at the same time. All right, here we are. So this is the lab. This is a positive flow fume hood. And so what they do in here is this is where they do all their sample manipulations. So like when they get their water samples, it pretty much just sits there in these specimen vials like these guys here, and it melts the water. But they have to have standards to compare them to. So they literally do the same sorts of things that we learn in class. Look, I made these earlier today. These are our standards, all sorts of different concentrations, so we can make a standard curve in order to be able to compare the concentrations of ions in actual ice. Next up, we're gonna see an actual mass spectrometer. All right, here we are at the South Dakota State University mass spec room. Okay, so this is a little bit of a complicated setup, but pretty fascinating. So this guy, I'm actually running samples of our ice right now. We're testing for sulfate. So let's talk about this real quick. So this guy is actually a liquid chromatographer, but we're not using that part of it for our samples right now. We are simply using it to grab our vials. So in here, so this is what I brought the vials samples down in, okay? And then I had to transfer them into that apparatus over there. So we're running 38 samples right now. It's gonna take about an hour, hour and a half or something like that. So anyway, so it's gonna auto sampling right here. And it's got a little needle, goes in, draws it up, brings it over here, and it flows through this tube, this yellow tube right here, into this, and this is our mass spectrometer right here, okay? Not very big, right? Okay, this is a special mass spec called a Q-trap, and what it does is it actually allows us to measure more than one isotope at a time. We can actually do 4. Q stands for quadrupolar. Okay, so you can do 4 isotopes all in one run. So you don't have to run it 4 different times. It's got a different, it can change the magnetism within it through electromagnets in order to filter out whatever isotope you're looking at. So anyway, so through this little tube, and I'll interject a video here, but you can see it. It goes right in there. You may or may not be able to see it in that video. Come out here, and then it goes through there, runs through there, a bunch of magnet sensors, and then it spits out a graph that looks kind of like this guy here. All right, so here's our graph that we get from our mass spec. Okay, so we've got 4 different ones we're testing for today. All right, and so the red one and the blue one are for the main isotopes of sulfate. Okay, remember that's what we're testing for, and you can notice that about between 0.2 and 0.4 seconds, that's when our sulfate hit the detector within our different testers. And then down here we've got a couple other isotopes that we're testing for and a couple other background noise in order to see what's going on. And so then from that data, you can grab the integrated peaks, you can grab the area under those peaks, and then throw them into Excel in order to start manipulating your data, not manipulating your data, but graphing your data and figuring out how everything goes from there. Be able to figure out, well, how did sulfate change over time 500 friggin' years ago? 500 years ago, we were able to see how much sulfate was dissolved in that ice. Super cool and just pretty awesome. So thanks for watching, and I'll see you on the flip side.